Howdy! This is our first lab report video on hand washing. We are Group 2, Ana Martinez and Hannah Garcia, Microbiology, Fall 2024. Introduction. The purpose of this lab is to evaluate the effectiveness of different hand washing methods with varying independent variables. In this experiment, hand washing methods were tested by using just DI water versus soapy sponge, each tested over different time sets. Significance and background information. The significance of this experiment is to prove which method is more effective in killing bacteria on our hands. It is also important to wash our hands before and after working with lab specimens during BSL-2 labs. As we may know, washing our hands with soap for some time has been said to be the most effective in significantly reducing the spread of bacteria and germs. So these are some of the materials that we use for this lab. 4 labeled agar plates, 8 sterile cotton swabs, liquid hand soap, sponge, saline, DI water, hair film, sealer, and a lab sink.
The methods for this experiment were after correctly labeling the 2 agar plates, dip a sterile cotton swab in saline and swab your right thumb. Then inoculate the R1 section with the cotton swab. Next, rub your right thumb and index finger together with deionized water over a sink for 1 minute. Then inoculate the R2 section with the cotton swab. Next, rub right thumb and index finger again for 2 additional minutes. Then inoculate the R3 section with the cotton swab. Next, rub right thumb and index finger again for 3 additional minutes. Then inoculate the R4 section with the cotton swab. Seal both of the agar plates with parafilm. After correctly labeling the 2 other agar plates, dip a sterile cotton swab in saline and swab your left thumb. Then inoculate the L1 section with the cotton swab. Next, scrub your left thumb with a soapy sponge for 1 minute and rinse off with deionized water. Then inoculate the L2 section with the cotton swab. Next, scrub your left thumb with a soapy sponge for 2 additional minutes and rinse off with deionized water. Then inoculate the L3 section with the cotton swab. Next, scrub your left thumb with a soapy sponge for 3 additional minutes and rinse off with deionized water. Then inoculate the L4 section with the cotton swab. Seal both of the agar plates with parafilm. Incubate all of the agar plates. Lastly, check on the agar plates and observe any growth. The results and observations that we got from our experiment were that in the R1 section, 1 colony grew. It was a Y isolated bacteria colony. In the R2 section, various Y isolated bacteria colonies grew. In the R3 section, 3 isolated Y bacteria colonies grew. There was no growth in the R4 section. In the L1 section, 1 isolated Y bacteria colony grew. In the L2 section, a few isolated Y bacteria colonies grew. There was no growth in the L3 section. And in the L4 section, it was the one with the most bacteria colonies. Overall, the bacteria in all sections are similar. In other words, there are no visible differences in appearance when we recorded our observations in our lab notebooks. Our conclusion, our experiment findings did not seem to have a clear pattern. The objective was to prove that washing our hands with soap for the most amount of time was going to have the least to no bacteria growth. However, our results might have been due to human error. The R results did not show to have improved by R4 as there was no growth. However, the L results were inconsistent, having no growth to little growth to no growth again, then having a lot of growth by L4. Some possible human errors that might explain our results include accidentally contaminating the sterile cotton swab, accidentally contaminating the thumb after rinsing but before swabbing, among other possible factors. Thank you.